Hi, let's talk about leukocytes. In this video, we'll talk about leukocytes, or white blood cells, the various types of white blood cells, their functions, and what information one might glean from a differential white blood count. So first of all, there are a number of means by which we can categorize leukocytes functionally and morphologically. So, for instance, one of the ways in which we can quickly group leukocytes is whether they are granular or agranular. So, granular leukocytes I remember as BEN, B-E-N, that's basophils, eosinophils, and neutrophils, which we can see here contain granules within them, and these granules can be released by the leukocytes to affect change. There are also agranular leukocytes, which are monocytes and lymphocytes, that are without these granules, so they appear more smooth on the inside. White blood cells can also be categorized whether they are phagocytic or not, and this process of phagocytosis means that they can engulf another cell. So, you can have this large, say, monocyte that comes upon a small bacterium, and then it can decide to engulf that bacterium and break it down, and then maybe present pieces of it to the immune system. There's also a phenomenon that some leukocytes are capable of, known as chemotaxis and emigration. And with chemotaxis and emigration, a white blood cell is capable of moving through a bloodstream, and then when it notices a chemical target, it can adhere to that target and then extravasate itself or move through the tissues in towards where it's needed. So chemotaxis is being attracted to or moving towards a chemical signal. So you'll recall this chart of various formed elements and their pathways of development. And in this particular discussion, we're really talking about these here. And so here are our granular leukocytes, and then here are agranular leukocytes. And you'll notice that all of the granular leukocytes are derived from myeloid stem cells, and most of the agranular leukocytes are derived from lymphoid stem cells. And the exception here are monocytes and cells that are developed from them, such as macrophages and dendritic cells and Langerhans cells, etc. They come from myeloid stem cells. Now, to be able to visualize cells and their contents, histologists will very frequently apply various types of stain, and a really easy staining combination is called H&E staining. H&E stands for hematoxylin and eosin. And this is a very forgiving staining combination that you can slightly alter the procedure and still end up with fantastic results. And it does visualize quite a bit of the cellular components very well. Now, the very basics of this stain, no pun intended, is that hematoxylin is the stain that adheres to basic structures, and it generally stains these structures blue. So in particular, some of the granules and the nuclei, where there's a lot of genetic material, these tend to stain quite blue or sometimes dark purpley. Whereas the eosin stain adheres to acidic structures, such as the cytoplasm, and stains them red or auburn, sometimes shades of pink throughout. And sometimes this will stain certain granules, but not to the extent that the hematoxylin will. And so we can begin to visualize and categorize and maybe even understand the etymology behind some of the names of these leukocytes as we move forward with this in mind. So let's take a look here at this blood smear, and as we can see with most blood smears, most of what we are visualizing here are the erythrocytes, and these erythrocytes stain very brilliantly auburn from the eosin dye. And we can also see some of these tiny little, blebs throughout. These are likely thrombocytes, but the most obvious standout cell in this smear is this one right here, and this is a neutrophil. Neutrophils get their name because they have no strong dye attraction. They are neither acidic nor basic. They are neutral. And so they don't really tend to stain very red or very blue. They're somewhere in between. You can see on neutrophils that they have segmented nuclei here. So there are very many lobes to them. Sometimes as few as 2, sometimes in upwards of 5 to 7. And as a result of this, oftentimes they're referred to as polys, which is short for polymorphonuclear leukocytes. Neutrophils can be found in both the blood and tissues because they're very good at chemotaxis and emigration, so that they can enter into the tissues from the bloodstream as needed. They have a very short lifespan, generally a shelf life of less than a week. And they are quite amazing generalists. So they're phagocytic, they're chemotaxic, and they're really good at degranulation. So these, as granular leukocytes, contain a number of both specific and non-specific granules. In particular, the lysozymes in these granules are very bactericidal. And so these are quite good at destroying bacteria. Now, neutrophils are the most abundant of all the leukocytes. So about 60% of white blood cells are generally neutrophils. And so they're the most abundant, and they're the first on the scene of an infection. Now, curiously enough, if you've ever experienced the phenomenon of having a little bit of purulence or pus draining, that pus, that aggregate of material, is dead conglomerations of neutrophils mostly. So we owe these cells quite a lot. Now, moving right along etymologically, we have the least abundant white blood cell, but probably it's important to mention that basophils get their name because they're so good at absorbing basic stain, the hematoxylin. And so they stain very brilliantly blue-purple, such as this lovely here. So this is the basophil. This other leukocyte is an eosinophil. There, so they're next to one another. Basophils typically have bilobed nuclei, and the granules, which are staining very dark, are very frequently obscuring those lobes of the nuclei. So you really have a difficult time making out the nucleus from the rest of the cell. Basophils, while they're not very abundant, have a very long shelf life. So generally they live between 1 to 2 years in the blood. They're chemotaxic, and so they can emigrate into tissues as well. They can degranulate, and when they degranulate, they can push histamine from their cells. So those granules contain histamine, which is a brilliant vasodilator, and heparin and serotonin, etc. Heparin's really good at anticoagulant. So these basophils are fantastic modulators of inflammation. They can draw a lot of blood to an area very quickly. They can also stimulate formation of IgE antibodies on mast cells, and this is the basis behind anaphylaxis, and they have other pathways towards anaphylaxis. So for those of you who might be unfortunate enough to be touched by a serious food allergy or another serious allergy, or know someone who is, usually basophils are part of the underlying mechanism for that. As a result of degranulating histamine and all of these other substances, usually basophils are found in higher abundances when a person is exposed either through environmental allergies, sometimes more specific allergies, or even has an ectoparasitic infection. So an ectoparasite is a parasite on the outside of the body, such as ticks or fleas, something like that. And these are the least abundant white blood cells. So to be able to find a basophil in a blood smear is really quite an accomplishment. Next up are eosinophils. Eosinophils absorb eosin stain, hence they're eosinophilic, which means that they are largely acidic on the inside. Their granules stain very auburn. The only thing that's really staining, basic on them are their nuclei, and usually they also have lobed nuclei, 2 to 3 lobes. The granules most often do not obscure the view of the nuclei, and that could be because they don't stain very dark. They have a very short lifespan, like neutrophils, usually less than 2 weeks. They have limited capacity for phagocytosis, but they are chemotaxic, so they can be drawn to an area, and they do degranulate. And when they degranulate, they can release histaminidase, which is going to break down histamine, and they can also degranulate substances that are going to modulate leukotrienes, which are other molecules that are important in an inflammation response. Another content of an eosinophil's granule is major basic protein. And major basic protein is pretty serious stuff for killing some smaller parasites. So endoparasites, blood flukes, aka schistosomes, are susceptible to major basic protein, and so an elevated level of eosinophils can sometimes be indicative of an endoparasitic infection. And these are also pretty rare, so only about 3% of white blood cells are eosinophils. So moving on to the agranular leukocytes, we have lymphocytes. So in this image, I can see but one lymphocyte. It really stands out here. Typically, the lymphocytes are staining blue or purple, and there are both large and small. This is a small lymphocyte. You can tell the difference usually because in small lymphocytes, the nucleus makes up the overabundant majority of the cell. I mean, I guess it does so also in a large lymphocyte, but you would see a little more cytoplasm in a large lymphocyte. So small lymphocytes, which are most common in blood smears, they're about 90% to the 10% of large lymphocytes, are inactivated lymphocytes. Now keep in mind, lymphocytes can be various types of T cells or B cells or natural killer cells, and under a light micrograph, they are impossible to differentiate from one another. The T cells and the B cells are involved with acquired immunity, so with some pre-existing exposure to an antigen, they're able to conjure up an immune response. immunity to a pathogen, whereas natural killer cells are innate. They don't require any exposure. They can just destroy. One thing that really separates lymphocytes from the granular leukocytes is that they are never phagocytic. Never, ever, ever. And these are pretty common. So about a quarter of all white blood cells are lymphocytes. Now monocytes. Monocytes are the largest of all of the white blood cells. They're pretty big. They stain blue, and one of their diagnostic characteristics is they have these puffy U-shaped nuclei. They don't have a very long lifespan in the blood, so usually fewer than 3 days, but if they do specialize and extravasate out into the tissues, they can last for months. Now, when they do move into tissues, they generally specialize, so they'll become more morphologically specific. And so, for instance, they can differentiate into macrophages. Macrophages are capable of phagocytosis, and these may either be fixed in the stroma of organs, or they may be wandering through tissues. Langerhans cells in skin are examples of differentiated monocytes. Dust cells in the lungs are examples of differentiated monocytes. Osteoclasts come from aggregations of monocytes. So these are really jack of all trades, or Jill of all trades types of cells. Dendritic cells are another type of cell that's a derivative of monocytes that can be found in either the skin or in mucosae. And these are important because they're antigen-presenting cells, or APCs. And what antigen-presenting cells do is they can engulf, they can phagocytize a pathogen, break it down, and then on the surface of the cell, they can present the portions of those pathogens, or the antigens, to parts of the adaptive immune system in the lymphatic system. So, for instance, they could present this to a T helper cell to help activate a cytotoxic T cell. And generally about 1 in 10 cells in a blood smear is going to be a monocyte. Now, differential white blood counts are really an art. I don't want to really go into the weeds here of anything that could cause an elevated or a diminished differential white blood count for any of these groups, but suffice it to say that an elevated level of white blood cells is called leukocytosis. And you can apply this to all of the different groups. So you can have neutrocytosis, lymphocytosis, monocytosis, etc., etc. A decreased level of white blood cells is leukopenia, and you can apply that to these groups. So you can have neutropenia, lymphocytopenia, monocytopenia, etc. So that's something to keep in mind. But I mean, you should know general things like neutrophils are elevated in bacterial infections. Generally, lymphocytes are elevated in viral infections, as are monocytes. For allergic reactions, you can see either eosinophils or basophils. Generally, eosinophils are more seen with endoparasitic infections, whereas basophils are seen with ectoparasitic infections. So those are some things to keep in mind. And that leads us to our assessment question, and that is, upon examination of a blood smear, you notice a field of erythrocytes interrupted by the occasional leukocyte. Which leukocyte would you presume to be the most abundant in the blood smear? Would it be A, basophils, B, eosinophils, C, lymphocytes, D, monocytes, or E, neutrophils? The correct answer is neutrophils. The relative abundance of leukocytes goes from neutrophils being the most abundant, lymphocytes being the next most abundant, monocytes being the next most, then eosinophils, and then basophils. And I always remember this as never let monkeys eat bananas. Thank you very much for your time.